Case Western Reserve University's Institute for the Science of Origins proudly presents the Origins Science Scholars Program. The Institute advances the scientific understanding and application of the origins and evolution of human and natural systems. The Origins Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and MediaVision. It's my pleasure to introduce Dr. Sarah Bagby, Assistant Professor in the Department of Biology at Case Western Reserve University. An environmental microbiologist, Dr. Bagby studies the interplay between the environment and the evolution and spread of microbe and virus mutations, which have the potential to shape and reshape the Earth system. Tonight, Dr. Bagby will talk with us about microbes and climate change. Please join me in welcoming Dr. Sarah Bagby.
Thank you all for joining me this evening. As Glenn said, I'm going to be telling you a little bit about the connections and the feedbacks between microbes and climate change. Now, at first glance, I know this may seem like an unlikely pairing. On the one hand, we've got organisms that might be a fraction of a millionth of a meter across. And on the other, we've got the whole world, nearly a couple of 100 million square miles on the surface alone. And yet what I want to convince you of this evening is that understanding microbial communities, and in particular, understanding their response to ecosystem change, is going to be an essential part of building the predictive understanding of climate change that's sorely needed to guide our own human response. Now, while I am a big fan of microbes, I am not going to try to convince you that, in fact, they constitute the entire world. I just want to show you that their role in it is an awful lot more significant than most people ever imagine. They are not, in fact, the whole world, as might be implied by my title slide, but they do perform as much as half of the world's photosynthesis. So I'm showing you here a map of global chlorophyll abundance created by a NASA observing program called CWIS. Chlorophyll, of course, is the pigment that's required for most of the world's photosynthesis. And you can see a lot of the world's photosynthesis is therefore probably happening exactly where you'd expect, in the forests and jungles and grasslands and agricultural lands of the terrestrial biosphere. But not all. There is also an awful lot of chlorophyll in the ocean, especially in high latitudes and in the equatorial band and around the mouths of the world's great rivers. And as I say, this marine chlorophyll is largely microbial, and those microbes perform somewhere between a quarter and a half of the world's photosynthesis. So if you don't find that compelling, I suggest that you try skipping every other breath for a little bit and see how you feel about that. Simply because they're not a part of our visual experience of the world in day-to-day life, people who aren't microbiologists tend not to think too much about just how much microbial life there is in the world. We think about the kinds of life we can see, mostly plants and animals. Microbes are pretty much the exact opposite of charismatic megafauna. They do not make a big showing in nature programs. So, for instance, my son is a microbiologist child and yet also a wildcrafts devotee. And when I ask him to estimate how much of the world's biomass is made up by these broad groups of living things, how much plant and how much animal and so forth, his first guess was that the biosphere is 16/17 animal. But no. In fact, the very large majority of biomass, about 82%, is plants. And remember, that includes trunks and roots and so forth. This is not just photosynthetic leaf surface area. But bacteria come in head and shoulders above everything else, nearly 13% of the biomass in the world. So tonight, we're going to be focusing on just a few of these sections that, aside from the pandemic, you probably don't spend that much time thinking about, though I realize that viruses have been more present in most of our minds than usual recently. So I want to place these for you in the context of everyone you know and love, as well as the 7 billion or so other people in the world. And you can see that pound for pound, there's more than 3 times as much viral material in the world as there is humanity. More than 100 times as much archaea, an entire domain of life that's newly enough discovered that it wasn't in my high school textbooks, and more than 1,000 times as much bacteria. So I hope you can see that what microbes do in the aggregate is liable to have a considerable impact on our world. And this has been true for a very long time. Photosynthesis was in fact invented by microbes more than 3 billion years ago. Eukaryotes just co-opted it by ingesting photosynthetic microbes and domesticating them into chloroplasts. Land plants didn't arise until a couple of billion years after photosynthesis was invented. And by that time, microbes had already dramatically changed the Earth's atmosphere, bringing CO2 way down and injecting molecular oxygen into the atmosphere with profound consequences for the rise of multicellular life. 3 billion years on, we have a global carbon cycle that looks something like this, with contributions from a mix of abiotic processes like volcanism and erosion, human activities like burning fossil fuels and deforestation and agriculture, and non-human biotic processes like photosynthesis. What I want to flag for you here is that a huge fraction of the processes feeding into the global carbon cycle are substantially mediated by microbes. Microbes contribute to photosynthesis, as we've seen, but also to the production and consumption of the phenomenally powerful greenhouse gas methane, to soil quality through nitrogen fixation, which outside of industrial settings is performed exclusively by microbes, and to the burial of organic carbon in the deep ocean, and so on. Everywhere you look, they are driving this cycle. But of course, it's not just carbon that microbes care about. Like every other kind of organisms we know of, microbes are built substantially of just 6 elements: hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. They also rely on a handful of important alkali and alkali earth metals: sodium, potassium, magnesium, calcium, and then some key transition metals that are important for crucial chemical reactions to be catalyzed. These 6 that I've marked in blue here, these are the biggies. And just as you might imagine from what we've seen of the carbon cycle already, the sheer abundance of microbes in the biosphere means that they have a huge influence on the global cycling of the elements they rely on. So you've probably all seen figures like these before in your middle school earth science textbooks, if nowhere else. What I'm showing you here, of course, is depictions of the global carbon, nitrogen, sulfur, and phosphorus cycles. I'm leaving out the water cycle, which of course is where you'd expect to find a lot of hydrogen and oxygen. Now, these pictures are important, and they do give a big picture view of how each of these elements moves through the Earth system. The problem is that when we look at these diagrams, we tend to extract a mental image that is a little like this, one where 4 cycles proceed independently. But that's not right at all. In fact, these cycles both enable each other and constrain each other. None of them moves independently. So really, the mental image that we ought to take away from these global cycling figures is a little bit more like this. One consequence of the way these global cycles are enmeshed with each other is that cranking up the activity of some cycles will tend to exert pressure on others. Equally, if there's something that limits some part of a cycle, that's going to impede the function of the other cycles too. So if we think about this in the context of the carbon cycle, for instance, why is it that the distribution of chlorophyll varies so much? On land, of course, we have some intuition for where we'll see less photosynthesis. It's typically going to be limited by water. And so in the planet's great deserts, right, the Gobi and the Great Victoria and Great Sandy Deserts in Australia, the Atacama in South America, the Sahara in Africa, the desert Southwest where I grew up, of course, we're not going to see a lot of photosynthesis there. In the ocean, our intuition is not so good, but I can tell you that these vast blue gyres of the ocean are essentially photosynthetic deserts, not limited by water, of course, but places where phosphorus and iron starvation limits microbial growth and photosynthesis. And you can see that the opposite is true in some places too. If you look at the deltas of the great rivers, you can see that inputs from weathering and continental runoff fuel photosynthesis at tremendous rates. It's not just the oceans where global cycles affect each other, of course. If we look at the continents, we can see broad global patterns in what's limiting plant growth where. These patterns seem to be driven primarily by climate, particularly patterns of temperature and precipitation and how those two relate to each other. Soil chemistry also matters. One of the big drivers is how much clay is in the soil. And it also matters which plants grow where and what balance of nutrients they need. But up here in boreal latitudes, there's essentially some sand in the nitrogen gear. And if you look at tropical and subtropical ecosystems, it's phosphorus that's mostly limiting plant growth. And of course, that means limiting how much carbon dioxide is being pulled out of the atmosphere by photosynthesis. So we can start to build a picture of how these cycles interact, driving each other or limiting each other in different ways. The trouble is that this picture that I've been building up for you is quite misleading because it suggests a fixed system. If you have a clock and you keep it wound and you keep it clean, it keeps on working essentially the same way. It doesn't go quietly about becoming a more competitive clock. But that's not going to be the case in a biological system. We've said that these cycles are linked because of the metabolism of living things that links them. So I want to spend a little while now talking with you about how that works and why that means that our picture of global cycles has to be dynamic rather than static. Okay, so I've been showing you these cartoons of big gears, and now here I am showing you a cartoon of small gears inside a cell. But where does this picture come from? A tremendous amount of work in the early and mid 20th century in biology labs around the world went into working out what we call reaction pathways, the series of chemical reactions that together convert some starting material or substrate into one intermediate compound after another, and finally onto some desired product. There's a lot that we still don't know, certainly, but the map of what we do know about cellular metabolism is really pretty humbling to behold. It's also totally overwhelming, so I'm going to show you just a small picture of it, just kind of one neighborhood on the metabolic map. These here are the reactions involved in making just 2 of the 4 major nucleotides, the small compounds from which DNA is built. Now, there's a lot going on here, so let me just zoom in a little bit for you. And now what you can see, I imagine, is that there is still a lot going on, right? It's tremendously complicated. Each of the arrows that you can see in this diagram represents a chemical reaction. Each of the boxed numbers represents a particular enzyme, the one or the ones that together catalyze that reaction. And the dots represent substrates and intermediates and products. One really notable thing about these maps is that you can see immediately the way the pathways are linked. Any of these compounds at the start of more than one arrow, that's what we call a branch point. If a molecule of that compound follows one metabolic pathway, great, but then it doesn't follow the other one. So there's a trade-off. If the cell needs that other product, that's a problem. Cells have to very actively manage how much of all these compounds is flowing through each branch of all these pathways. They have to acquire enough of the raw materials to allow the pathways to run. They have to make the right proteins that are needed for all of these reactions to run in the right ratios. They have to get the reaction products to the right place in the cell to do the job that they're needed for. It's enormously complex. And again, this is one tiny section of the whole map, just to make 2 compounds similar to the one that I'm showing you in the lower left-hand corner here. Even just in this section of the map, we can see the connections between nitrogen and phosphorus and carbon, right? This molecule is ATP, which we use for energy, and as I say, for our information-bearing molecules, our DNA, our RNA. And it has carbon and nitrogen and phosphorus and oxygen and hydrogen. It's got all of these raw materials. And the cell has to bring them together in the right ratios, in the right places, with the right chemical energies in order to create this fundamental unit of life. So it's easy to imagine how limiting the availability of one of these nutrients or supplying an excess of another might throw off all this carefully maintained balance. Here is our same wiggly cell cartoon. And the molecule that I'm showing you in orange is, of course, DNA. Right? So bacteria organize their genes a little differently than we do. In particular, they tend to have just one circular chromosome like this. And the central dogma of molecular biology is that DNA encodes RNA, a related but distinct kind of molecule. You get from DNA to RNA through a process called transcription. And then through a process called translation, we get to a totally different kind of molecule called a protein. And proteins are the real workhorses of the cell. They do, not all certainly, but many of the really important jobs. And so you might imagine that what you want to do as a cell is use every bit of DNA to make every possible RNA, and from that, every possible protein, right? Why else would you bother carrying around this DNA? But in fact, if you did this, it would be a horrible mess, right? Nothing would work right. All of that carefully balanced metabolic flow would be out the window. So instead, what you want to do is think about the genome of a cell as its toolkit, right? These are all of the tools that it has available to meet whatever challenges it needs to meet. You do not, for instance, walk into your workshop when you need to make a hole in something and say, aha, I know, a saw. That's the right thing, right? No, just like that, your cell needs to pick the right tool at the right time. So everything all the time is not the way. Instead, to have a well-ordered cell, what we need to do is use just the right bits of DNA, just the right genes, to make just the right amount of just the right RNAs, so that we can translate just the right amounts of just the right proteins. And the trick is that what is right in all of these cases depends on your environment. So these things change all the time. And the cell has to be very actively sensing its environment and modulating its response in order to keep on being a successful cell. So I'm going to show you just a very simple cartoon of some of the kinds of decisions that the cell has to make and some of the knobs that it can twiddle as it makes those decisions. So here is just a section of a chromosome. We've got two genes. One is purple and one is orange, okay? And they're going to be the right gene at different times. So at some point, it will be the right call for the cell to transcribe the purple gene. Biologists show transcription with this little hooked arrow. So that's what I mean when I show that. When you transcribe a gene, what you do is make an RNA, and then you can translate that RNA to make a protein. So sometimes that will be the right call for this cell. Sometimes, alternatively, we'll need to transcribe the orange gene to make an orange RNA, and from that, an orange protein. So the sorts of things that the cell might want to control in this scenario is, for starters, which regions of the DNA are getting transcribed. And there is an enormously rich literature on the sensory apparatus that bacterial cells and all of our cells and everything in between would use to make these kinds of decisions. You'd also want to control how many copies of the RNA that you've decided to make get made. Each of these RNAs can get translated independently. So broadly speaking, if you make more RNAs, you might imagine, well, then I'll get to make more proteins. But the copies of RNA don't last forever. Their lifetime in the cell can be actively modulated. You can make them stick around for longer if you want to make more protein per RNA, or you can degrade them actively if you decide that actually, no, it's time to switch over to making something different. Each RNA can get translated into, say, 10 proteins before it's degraded, or perhaps into 1, or perhaps into 100, or anything in between. So there are all kinds of little tricks that the cell has to modulate how efficiently each RNA gets translated. Once an RNA is translated into a protein, we are not home free. There is still regulation to be done. So sometimes a cell will covalently modify the proteins that it's made with different tags that either increase or decrease the activity of that protein. And those modifications can be reversed as the cell requires. And then, of course, just as RNA gets degraded when it's time to switch over to a different program, the same is true for protein. So the proteins might have different lifetimes, and the cell can actively modulate how long those lifetimes might be. So all of these processes, all of these decisions about which genes to express and when and how much and how long to stick with that plan are what we call acclimation. So the basic question in acclimation is just which of the tools that are available in my toolkit should I use now, right? This is what we call phenotypic plasticity. Genotype isn't changing, it's just what you do with it that's changing as you respond to your environment. And that's the sort of response that happens on a pretty rapid timescale, maybe as little as minutes, maybe lasting for as much as hours or even days. But that's not the only timescale that matters in biological systems. So we've been talking about our one wiggly guy, but of course, cells don't live in isolation. They live in communities consisting of a diverse range of other organisms. And lots of different things can be true of the members of these communities. So, for instance, some of them might be able to swim. And so you might have emigration of one population and immigration of another population. You might have some cells that can hold tight to the banks of a river or to grains of sand so that when there is a rush of water, they stay put while others get washed away. You might have cells with all kinds of different traits that allow them to become a greater part of their community or perhaps to fade out. The other thing that happens, of course, is that they're interacting not just with other cells, but with their environment. So, for instance, let's imagine that we have some leaf litter in a soil microbial community, right? This is a great source of nitrogen and carbon and phosphorus compounds. And so there are lots of degraders of leaf litter. So let's say that this blue guy can take a molecule from leaf litter and do some chemistry and convert it to a different molecule, probably extracting some energy along the way. And that different molecule can then serve either as a communication avenue, a signal, or perhaps as some food for a different kind of cell in the community. And let's further imagine that that signal or that food source allows this guy to thrive and perhaps outcompete somebody else. So all of these interactions, both between cells and across trophic levels and with abiotic factors in the environment, all of these are what we call assembly processes, where the basic question is who's there and how do they interact? How does that shape the total community? Assembly processes we would usually think about playing out over the course of seasons or years. In microbial systems, the timescales tend to be longer for larger, slower growing organisms. One thing that can happen, of course, as you start to build up larger populations of some of these organisms is that with more chances to reproduce, you get some more diversification. So I want to talk for a couple of minutes about how that diversification can happen. There are a few different pathways, and I'm really just scratching the surface here. So, of course, all of these cells have their own genomes, and probably the simplest method for introducing variety into a population is just plain old mutation, right? When a cell is going to divide, it has to make a copy of its genome, and sometimes that doesn't work quite right. And so errors get introduced. Most cells have proofreading mechanisms. Sometimes they're good, sometimes they're sloppy. The thing about mutation is that it's sporadic, right? This is happening at random, and so it might happen in any different part of the genome in any given cell division event. And so this can introduce variety into lots of different genes. When we have variety in genes, now that's a cell that has variety in its toolkit. And so perhaps it will respond to some stimulus in a new way. Another thing that can happen is that viruses can infect bacteria or archaea, and when they do, they can fundamentally change their hosts. So one thing that can happen is called lytic infection. And this is probably how you would picture infection if you don't think about it very hard, which is that the virus comes along and injects its DNA into the host, and then that DNA directs the production of new phage particles. And when there are lots of them, they burst the host cell, and that's a replication cycle. And that happens, and it's really important. For context, when I say really important, we know now that in the oceans, literally a third of microbial cells are killed every day by this process. So hugely important to cycling in lots of ecosystems. In soils, we expect, although we don't quite know yet, that a different process is going to be more common. That process is called lysogeny. And what happens then is that when a phage infects and injects its DNA into the host, if the host seems pretty healthy, and believe it or not, this is a thing that viruses are in fact assessing in microbial systems, if the host seems pretty healthy, then what happens is that the viral DNA inserts itself into the host genome and kind of hangs out. Now, the viral DNA often will have some genes that contribute to metabolism, even in the time before the virus starts reproducing and making more phage particles. So we have this lysogen, it's called, a host cell with a little bit of virus. And as it replicates, it will also replicate that virus and pass the integrated phage or prophage onto its progeny, where they can all do the little bit virus-influenced version of being that kind of host cell. Another thing that can happen is targeted diversification. So on first glance, this is going to seem a lot like mutation, but in fact, it is much more directed as a process. So this is a case where there's a particular molecular machine that can get used to focus enormous diversifying power on just one gene, so that instead of having a mutation here or a mutation there or a mutation in the other place, we have many different versions of just one gene. So I'm going to indulge myself here a little bit by getting into slightly more molecular detail, because this is a system that I've thought about for a while, and it still just blows my mind because we've only known about it for about 15 years, and it manages to encode more diversifying power in the space of about 1,000 nucleotides than the entire vertebrate immune system, which was the previous world record holder. So it's pretty impressive little system. So what I'm showing you here is called a diversity generating retroelement. In the black line, can you see my cursor, Patricia? Great, okay. The black line is just a section of the chromosome, and I'm highlighting a target gene in teal and an accessory protein, or rather a gene encoding an accessory protein in blue, and a gene encoding a reverse transcriptase in red. So what a reverse transcriptase does is take RNA as a template molecule and produce a new DNA following that template. So this is the opposite of the usual transcription process, hence the name. The way these systems work is that when the cell decides that it's time to diversify the target gene, it transcribes this little section here that's marked TR, or template repeat. So it makes an RNA, and the reverse transcriptase uses this RNA as its template and makes a DNA molecule on that basis. The trick is that it's a really terrible reverse transcriptase. And so on average, about 1 in 4 of the nucleotides in this new DNA are mutagenized. And so it's a very, very low fidelity copying mechanism. So now we have this new DNA molecule. And what happens next is that the reverse transcriptase and the accessory protein together carry that molecule over to the target gene and splice this new DNA into the target gene, replacing the original sequence. And so we've massively, massively mutagenized just one section of this one target gene. If you do the math on how big these regions are and how many nucleotides are getting changed, amazingly, one of these elements typically encodes the ability to make somewhere between 10 to the 20th and 10 to the 24 different variants, which is just an astonishing level of genetic change. And again, this is all focused on just one gene. Okay, so we've got all of these mechanisms of generating diversity in a bacterial community. One thing I haven't talked about yet is that microbes can also pass DNA among themselves. And when they pass DNA, of course, they're passing a tool. And that means that the microbe that receives it, if it keeps that DNA, can use it and change its phenotype as appropriate. It's not, though, just the case that you can pass DNA to other organisms like you. You can also pass it to other members of your community. And the effect of a given tool will probably differ depending on the genetic background that it's being added to, what other tools that new cell has available. So all of these processes that I've been telling you about are the raw material of adaptation. They are processes that generate diversity, and then all of the resulting cells have the chance to compete and to be selected by their environment. So here, the question is really, what would a better toolkit be? And can I build one? So I've been telling you about these three processes. We sometimes call them the 3 A's, of acclimation and assembly and adaptation, which all play out on different timescales. The really tricky thing is that they all feed into each other. If you acclimate successfully to some new change, you might have a better shot of making up a larger fraction of the community. You might be a winner in assembly. The particular collection of microbes that have assembled are going to determine what is the set of tools that are being passed around and shared. Which are the viruses that are successful and that are therefore then going to be inserting their DNA. If tools get passed around and shared, they then have the chance to be used for acclimation in different genetic backgrounds with potentially different effects. So all of these processes combine in a really complex, non-additive way. And the result is what we call emergent. What that means is that when environmental change comes, it sets off all of these complex, non-additive interactions, and the result is we really don't know. It's incredibly difficult to predict.
Some people seem to say that viruses are alive, and other people say they're not alive. Could you explain what viruses are and why they are or are not alive?
Right, sure. This is a question that always comes up. I think that it's not that important, actually. What viruses are, absolutely without question, is complex, dynamic, biological entities that evolve and that drive the evolution of cellular life. Sometimes, I know my textbooks did this, sometimes people will say, well, they're obligately intercellular. They can't reproduce without a cell. And that's true, but that's also true of some cellular pathogens. So for instance, rickettsia is a pathogen that can only reproduce when it's inside a cell. Do we think it's not alive? I think it's alive. It's a microbe. So it doesn't make too much sense to try to use that criterion to rule out viruses as alive. And I think particularly with all that we're learning now about the ways that viral DNA has been incredibly important over the history of life on Earth, and in the generation and spread of variation through the biosphere. It doesn't make sense to me to draw a line between the viral world and the biotic world as if they were two separate entities.
All right, there's another question here. How is the DNA passed among the microbes?
Oh, that's a great question. There are all sorts of ways. So I will try to not be encyclopedic in my answer because we'll be here all night. So sometimes the DNA exists as what's called a plasmid. So in addition to the main circular chromosome, a bacterial cell might also have other smaller circular pieces of DNA that can be passed through what are called pilus structures to communicate from cell to cell. One thing that we see over and over again is that, just like any other part of the machinery, cells can decide whether or not to make those pilus structures. And they tend to be expressed in times of stress, right? So again, you can picture somebody in their tool shop saying, I need something else. What I have is not working. Let me see what I've got, right? And reaching out to ask their neighbors, can I borrow that tool? So there's that. There is also a lot of DNA that is literally just floating around, right? Because other cells have died. And when they die, their membranes burst and everything that was in them comes out. And cells will take that DNA up. Again, they will do this more in times of stress and less otherwise. But, if you're starving for phosphorus, DNA is pretty tasty. You might take it up to break down, and you might find that actually it has something that's worth incorporating. Viruses are a huge route of transfer of genetic material also. I promised you a talk about microbes and climate change, and I've been talking about microbes, so I think it is time for us to get to the other part. I'm showing you here a map of the total soil carbon storage in the world. In this map, brown, more intense brown, is more soil carbon stored per unit area, and less intense is less. A lot of this map makes intuitive sense, right? If you look at the Sahara, as we've seen, there's not a lot of photosynthesis there, so not a lot of carbon is getting fixed into organic matter. So, of course, not a lot is getting stored. You might imagine that in jungles, we would see tons of soil carbon getting stored. And in fact, it tends to be more moderate. There's a lot of photosynthesis, but warmer temperatures tend to promote decomposition. And so not actually all that much gets stored. So instead, where we see the bulk of the storage is in northern latitudes, which stores somewhere between a third and a half of all the world's soil carbon. So here we've got lots of wet weather, which is great for photosynthesis, but it's cold, and cold inhibits decomposition. The trouble is that it's not going to stay cold. So I'm showing you here a map from the IPCC's RCP 8.5 scenario, showing what we expect to happen with changes in surface temperature. And the one thing I want you to take away from this is that it's going to get hotter faster in the north, in precisely these ecosystems that store most of the soil carbon. It's a little bit hard to think about what this might mean. So I want to show you some pictures now from the north, where these changes are quite literally earth-shattering. In the permafrost regions of the world, the ice that underlies the surface of the land is part of the structure of the soil. It's part of the fabric of the soil. You know, of course, that ice is less dense than water. That's why your ice cubes float in your glass. And when ice melts, there will be subsidence. Sometimes that means that the fabric of the soil simply tears. That can have effects on infrastructure, on transportation between remote communities, on buildings, on industry, and on people's lives as the ground shifts beneath their feet. I can't show you pictures like this of what the change means for microbes that are going to be as viscerally striking. But of course, it's going to be true that these changes are also profound for the microbial communities in the soil. So I work with a couple of groups of scientists who are trying to understand what these shifts look like for soil microbes in the global north. So we study a site in the permafrost zone. So here we're looking at end-on at the world, right? Permafrost is most stable, closest to the pole currently in what's called the continuous permafrost zone. And as you move south, it starts to get patchier, from discontinuous to sporadic, and then finally to just isolated pockets of permafrost here and there. So our study site is called Stordalen-Meyer. It's in the discontinuous permafrost zone in the thawing margin. It's an amazing site. So this is right at what's called the gate to Lapland, Laporten. And you can see at the upper left there why it's seen as a gateway. In winter, conditions here can be really harsh. This is not a gentle way to do field work, but it's an amazing site to be able to study. So at the upper right, I'm showing you an aerial image of the study site. And in the world of field biologists, this represents just a huge amount of infrastructure. I realize it may not look like that much from outside this small world, but what I hope you can see is these networks of boardwalks crossing the site so that it's possible for us to move around the site without disturbing the surface. We've got a couple of instrument huts and sheds that'll allow us to do some lab work there, and the whole thing is accessible by road, which is a big improvement over the helicopter that's required at most permafrost study sites. At the lower left, I want to show you a little bit about what's changing at the site. Right? So this is a boardwalk, and if you build a boardwalk, typically you want it to be dry. That's kind of the point of them. And this one was until it wasn't. So this is a site that's seeing rapid change, and we are doing our best to chronicle what's happening and understand how the biological processes I've been telling you about are feeding into the outputs that this ecosystem will produce. So one of the things that is incredibly useful about this study site is that it's been a research station for more than 100 years. So we have deep records of climate at the site. And even beyond that, we've got the traditional knowledge of the Sami people who have their own narrative stories of changes at the site over a longer time than that. The Meyer has been studied intensively for about 50 years, and there is in-place instrumentation that allows us to do really remarkable things, like, for instance, get a nearly continuous stream of recordings of which gases with which isotopic compositions are being released by the plant and microbial communities of the Meyer. We also have in our collaboration Mike Pallas, who's a remote sensing expert. His drones allow us to look at the plant composition of this site down to 3 centimeter resolution, which is astonishing. So what we see in this site is a progression from the intact landscape, which is called a pulsa habitat, to a couple of more thawed states. So I want to just take you on a bit of a photo tour of what these different habitats look like at the site. So this is pulsa. This is the sort of the baseline state for this site. I'm showing you in the upper left a bit of what the vegetation looks like. It is a diverse plant community. This white object here is one of our sampling quadrats that we use to just count how many plants do I see of which types and how mixed is that community. At the right here, this is a close-up of what the surface of the pulsa would look like after we have taken a soil core for a sample. And what I want you to notice is that this is a dry landscape. So at the pulsa, the underlying permafrost is intact. It has not thawed. We can pull out these soil cores. We can section them and sample them and analyze them 16 ways to Sunday. We can put bits of the soil into incubations and measure over time to see which gases they produce and how those communities change when we poke them in different ways. We can also move to a partially thawed region of the site, which is a bog habitat. So here you can see, I think, that the plant community is getting less diverse. There's a lot of sphagnum moss at this type of site. You can see, I think, just how wet that moss is in the hand of this member of our field team. And if we pull cores here, what you see is that there's a puddle left behind. So the bog is, this always strikes me as a little funny. You don't think of a bog as a really dynamic environment, but it is. It's hugely dynamic because the water table is moving up and down with rainfall events. And so that brings with it lots of big changes for the microbial community at the site. If we move all the way across the thaw gradient to a fen habitat, what we see is that there's standing water pretty much all the time. So the plant community here is still less diverse. It has a lot of sedges, which matter because they facilitate gas transport in different ways between the atmosphere and the soil. And if we pull a core here, we can barely even see where the hole was, right? It's immediately full of water right up to the surface. So just to zoom out a little bit, this is how we picture the thaw gradient across the landscape, from intact pulsa to thawing, partially thawed bog, to fully thawed fen. And we can take lots of measurements as we move across this thaw gradient. We can measure how much oxygen the microbes living in the soil in these different habitats have access to, what the pH is, what the temperature is as we move down through the depth horizons within the soil. And what I hope you can see in all of these plots, which are taken by the Isogeny Consortium over the last decade or so, is that there are big changes. The pulsa microbes have access to oxygen. The bog and fen microbes, well, fen basically not ever, and bog it's variable. It depends whether you're above the water table or not. The reason I mention this is that having access to oxygen is an enormous constraint on the kinds of chemistry that it's possible to do. And so you might expect to see completely different metabolic outcomes in an anoxic versus an oxic environment. pH also makes a big difference. At more acidic pHs, you might have simply different sets of organisms, or some of the same organisms might need to put a lot more energy into maintaining their internal pH and have a lot less energy available to grow. And of course, temperature matters too. Temperature matters a lot, for instance, to the membrane structure of cells. If you have ever tried to wash butter off your hands in cold water, you know that lipids respond strongly to temperature. If you have a membrane that is nice and fluid in 20-degree weather, it might be too fluid in 30-degree weather, and it might be brittle in 10-degree weather. So here's a site where we're seeing a lot of environmental change. And we know that that is going to feed into processes of acclimation and assembly and adaptation. And what we really want to know is what happens next. Because as we've seen, there is so much soil carbon stored here that what this microbiome does is going to be either an important sink for carbon from the atmosphere or a really terrifying source. And at this point, because these processes interact in such complex ways, we don't actually know which one. So I want to show you not that full carbon cycle image, but a sort of soil eye view of the carbon cycle for the permafrost. There are lots of different processes that we might care about here: photosynthesis, respiration, fermentation, different ways of making and of burning methane. And in particular, we want to pay attention to places where methane gets released from the soil. So methane is something like 28 times more potent as a greenhouse gas than carbon dioxide, pound for pound. And so if we have an ecosystem where a lot of methane is getting released, that is going to be a big source term in climate modeling. One thing that can happen is that carbon dioxide can get pulled from the atmosphere by photosynthesis and then converted in the soil to methane and then released, which is not what we want. But it's even worse if what happens is that ancient carbon that has been safely locked away in the soil thaws and then gets pushed through the same pathways. We don't know what the shifts among these pathways will be. It's possible that photosynthesis will thrive as the plant community changes. It's possible that the methane burning pathways will thrive. But we don't know what the balance will be between these different fluxes. And so we don't know what to expect the outputs from this ecosystem to be as we throw all of these changes at it. So we're trying to study this in a lot of different ways. These are some beauty shots from my lab. We've got microbes that we're trying to isolate so that we can grow them in the lab and poke them in all kinds of ways and figure out what they do. We want to understand their baseline physiology and then also how they respond to changes that we know are happening at the site. We're also interested not just in which genes they decide to express when, giving rise to which new kinds of cycling inside the cell, but also into changes in the way that they interact with each other. And these are things that we need to study really in situ at the site, because the community is so complex that as soon as we remove just a subsample of it, we're asking a different question than we can ask in situ. So we need to be thinking about how all of these different kinds of organisms are exchanging metabolites and are exchanging genes and are changing their toolkits, retooling as needed to meet the challenges that a changing, thawing permafrost landscape is throwing at them. It is not going to look like a clock, but we may be running out of time to figure out what it does look like. So with that, I will put up the many, many, many names of the enormously large teams who are working on these very complicated problems. The collaborations that I've mentioned to you are the Beer Soil collaboration and the Emerge collaboration, who are supported by the NSF and the Department of Energy. My lab also has support from the Expanding Horizon initiatives at Case Western Reserve to do some work on developing model systems of these soil communities. And with that, I will leave you with this picture of a boardwalk at the site and some gaps in our knowledge of the path ahead.
One question is, how is the carbon stored in the soil? What are the chemical components?
Oh boy, that's a great question. It's incredibly complex, right? So a lot of it is just plant matter, just litter, right? So the stems of the plants that have grown before and died, the leaves, they all settle down and they get compressed beneath winter snow. There's sedimentation and they wind up just persisting in this landscape. It's a little, if you've ever seen a bog body, right? These are landscapes that preserve. So there are, I'm going to get the era of human history wrong, Patricia, you can correct me, Paleolithic, possibly Neolithic, people who are preserved essentially intact in bogs. And these environments can also preserve plant matter incredibly effectively. In fact, it makes our life pretty difficult when we go and try to sample. You might have a picture in your head of us going out with all of these very high-tech tools to do our sampling, and that is partly true, but also we bring bread knives because we have to cut through really dense, thick tangles of plant material.
That would be the Neolithic.
Okay, thank you.
We have several people writing in to admire what a great talk it is and what a good job you've done explaining complex systems and especially the diagrams and everything. We'd had a question earlier. When was the process of acclimatization first discovered?
The answer depends on whether we're interested in molecular mechanisms or interested in phenotypic observations, right? So people have known for a long time that organisms change what they do in response to what they experience. And in biology, and in microbiology specifically, this probably goes back approximately as long as people have been making bread and beer, right? We can tell that there are changes in what organisms do when we feed them in different ways or heat them to different temperatures. But as far as having a molecular understanding of how those changes are happening, we are still actively discovering new ways that microbes twiddle all of these different control knobs. Certainly, no one had a grasp on it prior to the working out of the central dogma in the 1950s and 60s. But I think the most recent new mechanism I saw was published in 2018, and I wouldn't like to bet that there's been nothing else new since then.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.